So, what I thought I'll do today, as the title shows, is to give an overview of some of the work that we have been doing in my lab on using what are called as single molecule sequencing approaches to map modifications in RNA. Okay, and in the a little later part of my talk, I'll be talking about how you can use these ideas to map the secondary structure of RNA, and then talk about how RNA-RNA interactions can also be mapped using this technology. So this would be largely a technology kind of a talk where we are trying to showcase our efforts in the last few years on an experimental and computational front on trying to work with new technologies. But before I get started, thank you. Thank you all for coming, and thank you, Katie, for hosting me. Uh, let's see this one. Clicker. Okay, the clicker doesn't work. The focus probably. Yes. Oh, I did something. Okay. Okay. Before I get started, let me give a brief overview of the field of RNA modifications and the recent developments in RNA modifications, which is why we have been excited. So, RNA modifications are broadly referred to as the chemical alterations of regular nucleotide bases in RNA. Most of you have heard about the idea that RNA has four nucleotides: uracil, cytosine, adenosine, and guanine. Now, what we have noticed in the last, I would say, 20 years, but much more in the last 10 years or so, is that each of these nucleotides can be converted into other nucleotides. For instance, adenosine, based on your textbook knowledge of high school, you can say that there is a change from adenosine to inosine. And I've marked in red color the change that has happened. So this happens in the cell with a class of enzymes called ADARs, and there are some colleagues right here in Bloomington who have been working on ADAR family of enzymes which convert adenosine to inosine. Likewise, uridine can be converted into pseudouridine, which is simply a flipping of a double bond from one location to the other, and this is done in the cells using enzymes called pseudouridine synthase. Okay. Now, there are other modifications like N6-methyl adenosine, N1-methyl adenosine, and I've highlighted what their chemical structure looks like. These are small changes which can change the nucleotide. We are talking about individual nucleotide on the RNA molecule. Now, we have now accumulated in the literature about 160 modifications that are naturally occurring in life forms. So you may be wondering, why are they interesting? For starters, the reason why the COVID vaccine was successful is because Moderna, for instance, replaced all the uridines, all the use on your vaccine into 1-methyl pseudouridine. Okay, so pseudouridine is the modification that you see. Pseudouridine is this particular nucleotide. Okay, uridine to pseudouridine is simply a double flipping of a double bond, and you add a methyl group here that becomes 1-methyl pseudouridine. And in COVID vaccine, this 1-methyl pseudouridine was used to replace all uridines, and that increases the immune rejection, so decreases the immune rejection to humans. Now, if one modification can be used to create a vaccine, you can imagine what kind of functions that you can create by studying all of these different modifications that are existing. So, what do we know about these modifications so far? So most of the studies that have been done in, let's say, last 15 years or so have been on M6A, which is this particular modification. It's also called N6-methyl adenosine. That is the methyl group on the end position, or A2I, which is adenosine to inosine. And then pseudouridine, which is the conversion of uridine to pseudouridine, or M5C, which is this. These are the four modifications that have been extensively studied. And so, what else do we know about this modification? So the other aspect that has been coming into picture was, if you take an mRNA molecule, mRNA stands for messenger RNA. In a messenger RNA, the historical notion was you can see one modification and you can see another modification and you can study. But if you know that these modifications occur in multiple locations in a given RNA molecule, in a given mRNA molecule, for instance, what is the crosstalk between these modifications? How do these modifications contribute to the linear or structural aspects of this RNA molecule is a big and open question. Okay. Now, the other component that has been exciting in the field is some of this modification can result in a complete base change. For instance, adenosine to inosine is a complete base flipping modification. A cytosine to uridine is a complete base flipping modification. But the majority of the modifications are simple alterations, just a methyl group, a double bond change, hydroxyl group, or the backbone of the RNA gets attached with a methyl or acetyl or some other group. So what is the role of these modifications? Sometimes if they flip the base, yes, we know that that is changing the stoichiometry of how RNA-RNA interactions are happening. So we have been excited about this field way back in 2017 when I started getting into wet lab space at IU. And but at the same time, there was some literature talking about these modifications are reversible nature. Okay? If you have heard about the concept of epigenetics, epigenetics has been largely shown as sort of not always reversible. Most of the time, it's not reversible. But in the case of what people are calling as epitranscriptomic code, it's been shown to be reversible nature. So let me elaborate what I mean by a reversible nature. When you have an RNA molecule and you have an adenosine, this adenosine can be converted into methyl adenosine. And this happens with the help of what are called as writer class of enzymes. These are enzymes, metal enzymes. All these enzymes that you are seeing here are enzymes in human which are able to convert adenosine into M6A, and they're called as writer class. Okay. Now, what has been shown also is this M6A can be modified back into adenosine through a class of enzymes such as FTO, ALK-PH5. These are called eraser class of enzymes. Okay? This reversible nature is interesting. What is even more interesting is when you have this modification, attachment of let's say simple methyl group, the outcome of this RNA molecule, the fate of this RNA molecule can be very distinct. Now, some group of proteins which bind to RNA can target it for translation. Some can dictate the stability of the RNA. Some can dictate the splicing and nuclear export and so on and so forth. So the functional outcomes could be very different depending on the modification. Now, this idea of writer, reader, eraser model was first shown for M6A modification. Now, it is up to our imagination whether these readers and erasers are existing for all the 160 modifications that are reported so far. Because even for the writers, we don't know which enzymes are able to do this modification in humans and other systems. So it's a big open question. What are the players which are dictating this epitranscriptomic code in the systems? So we have been interested in this space also because there are a lack of technologies. So most of the time, when you have these modifications, groups end up using one of the two approaches. The first type of approaches are antibody-based approaches. In antibody-based approaches, you use an antibody against a modification. So this antibody goes and specifically binds to this, and then you cleave the rest of the RNA, and only the protected fragment is then sequenced, okay, in antibody-based methods. But having an antibody specific to the modification is very important for the antibody-based technologies, okay, for mapping the location of modification. The other type of methods are the chemical methods. So, for instance, if you wanted to map M5C, methyl cytosine, you would end up using sodium bisulfite. So, what does sodium bisulfite do? It converts all the cytosines in the RNA into uracils. It changes all cytosines into uracil, but except the M5C. The methyl cytosines are not converted into uracils, but they're kept as cytosine. So when you sequence the whole RNA of a cell, you can see that only Cs are corresponding to the methyl cytosine. It's a simple idea, but the challenge would be the exposure time of sodium bisulfite. Have you converted all the cytosines, for instance, right? If you have not efficiently converted all the cytosines into uracil, then you're going to start having false positives, okay? So there are these drawbacks. But more importantly, all these methods can only study one modification at a time. You will have to do one assay in one cell line using any of these to study. So if you wanted to identify this and this, you have to do this chemical assay, and you have to do this antibody assay to map M6A and M5C. And most of this is still happening in short read technologies, which can only identify local regions, not give a context, a phasing of which modifications are occurring in a single molecule resolution. Okay. So what else is happening? In terms of diseases, this chart shows you what are the diseases. The diseases are marked in red. All these modifications in red have been documented to be associated with some disease. Now, the number of modifications for which techniques are available are shown or circled in green. You would see that there are very few modifications for which detection techniques are available, although even more so, the number of modifications have been documented to have disease relevance. Okay, so this suggests that most of these techniques are only able to detect one modification. They have typically higher error rates and low resolution. Low resolution meaning that the context, the phasing of these modifications are not. They're only anyway available for few modifications. So few years ago, we started developing an internal database because we believe in, if not gold, at least silver standards of data to build models. So what we said was, what is said in the literature? How many different modifications are actually reported? So we were able to collect some 3 million sites in human and mouse genomes, which have been reported by various studies, about 50 different studies, on where these modifications are occurring. And this is corresponding to some 11 different modifications. So this database allows you to search by gene, by coordinates, by modification type, and you can visualize the locations, nucleotide resolution locations of where these modifications are occurring. We integrated with the human gene expression data, not a single cell, but bulk sequencing in human and same in mouse to see which tissues are these modifications likely to be active. It's a simple integration to develop a genome browser for our internal understanding of where these modifications are occurring and how we can use this existing data for development of our models. So you may be wondering, so how exactly are you planning to identify these modifications? So this brings me to the notion of technology development. We have bumped into the nanopore sequencing, which I will elaborate in a minute. But this has enabled us over the years to develop a number of tools, Raven, Penguin, and NM Nano. I always like birds, so I started naming, and then one of my postdocs said, nah, I don't like birds, so I'm going to give a very traditional kind. So we had a disruption when we started building tools. So, but we may go back to adding more bird names for these tools that we have been building. So each of these tools that have been, for instance, this has been built for predicting M5C. This has been built for predicting pseudouridine, and a lot of industries interested in pseudouridine prediction. So Penguin is popular. And NM Nano is for predicting backbone modification. So I'll elaborate what goes behind this. So before I get into how exactly these data sets are generated and how we build models for, let me talk a little bit about nanopore sequencing. So in nanopore sequencing, when DNA or RNA molecule, in here you're seeing DNA, a double-stranded DNA, a single strand of DNA, as it moves through the pore, there is a characteristic disruption of current. Okay, when a molecule moves through, there is a characteristic disruption of current, which looks something like this. So you have a time versus current captured, and this current should be able to help us to predict what are the nucleotides that are passing through the pore. That's the idea. So when DNA sequencing is done, it is done at 450 base pairs per second. That's the speed at which the DNA molecule moves. For RNA sequencing, of course, you won't see double-stranded. You'll see single-stranded RNA going through, as opposed to single-stranded DNA. That is read at a speed of 80 to 100 base pairs per second. A typical experiment runs from 24 to 48 hours. And you may be wondering, if one pore sequence at 80 base per second, how much data are you going to get? Well, there is not one pore. In a typical flow cell, you have about 2,000 pores working in tandem. Each pore, it's like a factory in itself, running the molecules and getting these currents. So you are connected to a GPU. Now, these devices, you will see one of those. I have one in my office. I always fancy showing to the students, which doesn't work. That's why it's in my office. So these devices enable you to generate about 1 million reads to 2 million reads on a desktop device. But you want to scale up You can put 10 of these devices together and increase the throughput, which are, of course, more expensive. Now, we have used it during COVID to do virus detection because viruses are small. You can do real-time, on-field kind of diagnostics by pooling samples and so on. So that's some background about nanopore sequencing. So, well, most of the sequencing on nanopore has been on DNA. And we want to sequence RNA as an RNA molecule. In fact, any sequencing that you have heard about ever is always about sequencing DNA. Even if it is RNA, you convert RNA into cDNA and then you sequence. When you convert RNA into a DNA, all the modifications essentially are lost. Okay? And which is why we are excited about this technology, because you can sequence RNA directly. That's why it's called direct RNA sequencing. And the library preparation sequencing is about a 2-hour process. A first-year undergraduate could create this library and start sequencing. So typically, you would isolate mRNAs with poly A tails. You will use oligo-DT primers, and this oligo-DT would stick to the A's because of the complementarity. And there's an optional reverse transcription step. The reverse transcriptase would start elongating it, because if you don't do this step, the RNA becomes sometimes unstable, doesn't go through the pore. So to stabilize, you do a reverse transcription, a little bit of it, and then you attach the sequencing adapters, and then it runs down the pore, and the blue molecule is what you're essentially sequencing. Okay. So we have used these Minions. These are devices from Oxford Nanopore Technologies. We have sequenced number of human cell lines and even mouse, just for fun, mouse brain regions to see whether we can do it. There are issues. We can talk offline about what kind of issues you would see on primary. Now we have been collaborating with some oncologists to sequence metastatic tumors to see whether we can identify changes and so on. So coming to the data. What this plot is showing is in HIP-G2, which is a liver cell line, human liver cell line, the distribution of read lengths. Okay, each of these reads is a single molecule. Now, typically in short read sequencing, you would probably get 200 nucleotide fragments if you're lucky. Okay, but look at the distribution. Your median length here is about 1,800 nucleotides. So your mRNAs that you're talking about are at least 1,800 nucleotides long in a cell line, in HIP-G2, in a liver cell line. K562 is the other cell line. It is a leukemic cell line. And we found that the length of the fragments, these are single molecule, end-to-end, from 3', 5', you are sequencing about 1,200 nucleotides long mRNAs that we have been able to identify. These are truncated just so have a distributions comparable. But of course, you can get as long as the length of the transcripts. So this is what I mean by single molecule sequencing and sequencing RNA as RNA. So what next? Well, there's a lot going on in this slide, but the main message I want you to take is you can sequence DNA or RNA. When a molecule moves through, you capture the current. When you do RNA sequencing, at any given time, there are 5 nucleotides. Again, this is cartoon, so it may be a bit exaggerated how many nucleotides are in the pore. But in a real runs, you have 5 nucleotides in the pore. These 5 nucleotides would be having a current something like this. Okay. And you would plot current in amperes, picoamperes versus time. This is the real data that you're getting. So if you wanted to know from current what's the signal or what's the nucleotide, you have to train for all possible 5-mers, right? All possible. First of all, you create synthetic molecules with known sequence. You run them down the pore, get the current, get the sequence, and try to build models. This is what the company has done for the 4 regular bases, AUGC. But what if you are interested in a methyl cytosine, right? Instead of a C, it's a methyl cytosine. You could say, well, I will create a synthetic molecule with a modification, or I know that these are the locations in the genome where a modification is occurring, so I can extract current for these and try to develop models, right? Machine learning, deep learning, whatever. So in the early stages of our efforts, we noticed that for DNA, there are 2 different tools, Tombow and Nanomod, which are visualizers for seeing the DNA signals, okay, current signals. But for none were existing for the RNA molecules, okay? And we also noticed that the company itself published a paper showing that if you sequence a molecule like this fragment with an unmodified A and a modified A at the respective location, there's a tilt in the current, okay? This is the location. And you're seeing that the current in the unmodified is higher, but when you replace that A with an M6A, you see the current to be. It's a very subtle, but if you can capture that using machine learning models, you can start mapping this modification. So we started developing a visualization tool with a colleague, Kairi Reda, on our in Luddy, where we said, like, can we plot these k-mers? We call these k-mers, where k is equal to 5. What's the current looking like, and can we do dimensional reduction, whether there are modified and unmodified composition of this data. And we started seeing that, yes, you can use this. You can start seeing differences in the current, and you can attempt building models. So we then said, OK, let's target one particular modification, cytosine to methyl cytosine. And for this particular modification, there was a lot of work done, okay, using bisulfate sequencing. If you remember, I said, bisulfate sequencing is used for mapping a methyl cytosine. And two different studies independently reported good quality data, okay, for the cell line called HeLa. Okay, HeLa cell line is, we love working with cell lines because you can tame them very well in the lab, and we know what to expect. There's not many variables that you have to work with. At least for technology development. So an additional aspect that is interesting about these modifications are dynamic, okay? What it means is, if you take an RNA molecule, and because a copy of an RNA molecule is not one, there could be 10 copies, 20 copies. Not all RNA molecules get modified. There's stochasticity, okay? Let's say a modification occurs at 50%. What that means is, if there are 20 copies of RNA molecule that are generated, only 10 of them have a modification. So that makes the problem even more complicated or interesting, depending on how you look at it. So using short-read data, we were able to show that majority of the modifications occur at a low probability of about 20%, okay? While about 60, those modifications which occur at 60% are probably trustworthy, because you are not doing deep sequencing when you do nanopore sequencing, because it's an early stage technology. So that was one of the additional things that we had to take into account. Together with Epitomy, which is a database that we put together, we were able to generate a list of all bona fide locations in the human genome where modifications are known to occur, okay? So you have a gold standard, so-called gold standard locations where modifications are occurring. And you have a technology where you have sequenced, and you have a reason to believe that change will occur when this modification occurs, okay? So this brings me to the experimental data generation. You do a library preparation, mRNA extraction, sequencing through direct RNA sequencing protocol, do a base calling. Base calling is the step where the onboard base caller gives you the 4 regular bases AUGC. And we noticed that about 2,500 locations in the genome had various probabilities of this M5C modification, okay? Because we know location where other short-read data has been supporting modification. So we take this data, and again, anytime you deal with these new technologies, there are new file formats, all kinds of challenges that you would see. So, and then we extract this current signal. About for 20,000 locations, we could extract signal. We used it to develop machine learning models, okay? That's the idea. And then you validate it, okay? Because you could do 50-50, 80-20, all kinds of splits to develop these machine learning models. So before I get into the modeling, you may be interested in, is there a change in current? Well, this distribution shows the current values for this 5 nucleotides when there is, this is unmodified, this is modified. The modified cytosine exhibits lower current, while the unmodified exhibited higher current. And this is not for nucleotide. Remember, at any given point in nanopore sequencing, 5 nucleotides are in the pore. The middle nucleotide is of your interest. So it's a sliding window. The first 5, second to 6th base, and so on as the one. So you have to extract the current for the position that you're interested in. But the reason I say position of interest is others have told there's a modification here from other studies. So use that coordinate to pull the current for that particular region. So we played with a whole bunch of machine learning and deep learning frameworks. And we noticed that as the probability of modification increases, our models start getting better, okay? Obviously, it's because the data is noisy at lower probability of modification. So you're not having a good discrimination between whether it's a modified or unmodified. But when you say 70% of the time it is modified, the data is cleaner, right? So that's why the models start getting better. So we played with a number of these, and then we realized that just current alone is not sufficient, because sometimes as the molecule moves, there is a pausing. So the time elapsed is important. how long is the signal is important. So there are other parameters that are important, and that kept us busy for some time. How can we start using all these features to improve our models? So that's why our early models are like 60, 70% max. And oh, did I something wrong? So as I said, like, the challenges were like the drawbacks like false positives, less signal data, experimental approaches, not too many complementary approaches were available, because if bisulfite data itself is noisy, so you're comparing not against a gold standard, you're comparing against, I don't know what kind of a standard, right? So those challenges do exist. It doesn't go forward. So this is one study where I'm going to quickly summarize. So we used HEK 293 cell line. This is for predicting pseudouridine. We have locations where other groups have reported where the pseudouridine modification occurs. We extracted the signal on a global scale for the modified and unmodified uridines, right? Unmodified would be uridine, modified would be pseudouridine. We extract features like sequence, signal statistics, develop machine learning models. We've used a whole bunch, okay? Support vector machines, neural networks, deep learning models, and so on. And when we build these models, we often use a second cell line for validation. You use one cell line for training, other cell line for validation, okay? And we ended up hitting like more than 90% accuracy for training and testing. So you may be wondering, maybe the sites that are seen in one cell line are the same sites that are seen in the other cell line. No, that's not the case. So this Venn diagram shows about 55,000 sites detected in HEK, but about 40,000 or so, 45,000 or so detected in HeLa. There is some overlap, but not a lot. So training on one, building models on one, and testing on the other is a good model to say that the models are reproducible. Interestingly, when you compare this at the gene level, so this is at the nucleotide resolution, right, where the modification is occurring in two different cell lines. But when you compare with the genes which have modifications in one cell line versus the other, the overlap gets better, which means a gene can be modified in some locations in one cell line, and it could be modified in other locations in the other cell line. So we don't know what is happening. And sometimes I even question whether our sequencing depth has something to do with that, but that's for the future. So when we plot the distribution of this modification, so these are called topology plots in the field of epitranscriptomics. The x-axis is showing 0 to 1 as the length of the gene, okay? Each gene will have different lengths. So 0 is the 5' end, 1.0 is the 3' end, and y-axis is showing the density. We notice that most of the modifications happen in the 3' end, in both the cell lines, and this is not just true for pseudouridine. You will notice that even for other modifications typically happen in the 3' and 5' ends of genes. So moving on, we try to use a different approach because the reviewers were not happy with training on one cell line and testing on the other cell line. So they asked us for this 2' oxygen methylation, which is another modification which is important for stabilizing vaccine. The first one I was talking was the pseudouridine. The other one that you see in COVID vaccine is 2' methylation, okay? So for this modification, we said we'll do two cell lines, HeLa and HEK. We do 50-50, 50% for training and 50% for validation. Again, there is a group in Chicago who are who's who in the epitranscriptomics field. They published a chemical method for mapping this modification. It's noisy. There was a correction to that paper. That's a different story. But we used high confidence sites that were reported in that chemical based method. And we said we'll use 50% of the two cell lines data for developing models. So we did a random forest model, an XGBoost model, and build the model. And then use the remaining 50% of the two cell lines to validate. This is a different approach than opposed to compared to the first one, because the reviewers didn't like one cell line training and one testing on the other. They said, okay, take it, we're okay with it. And then it actually yielded even better accuracy. And the reviewer could say that remove it because it's looking better accuracy than the previous one. And we know why it was, because we are training on both the cell lines, so it has seen enough variability, so it was doing even better than. Again, the distribution of this modification is very typical. Modifications occurring mostly in the 3' and 5' ends, okay? So other aspect that I have always been fascinated was synthetic biology, or trying to design molecules. So we have tried this, and we have some successes and some challenges. And so what we were trying to do was, can you put this regular nucleotide like guanosine and modify nucleotide? So if you recall, NM is where N is any nucleotide. So when you put a methyl group on the backbone, I'm sorry if I'm bringing a lot of you to the chemistry, but when I got into this, I lost a lot of my chemistry because the last time I picked up chemistry was in undergrad. But when you're passionate about solving something, your students and will make you to learn. And I was picking up all this, yeah. I was very good in chemistry, but I lost a lot of it over the years. So this is a 2' methylation, okay? But this is on a guanosine. But you can, if it is a cytosine 2' methylation, you call CM and so on. But if it is any base where a methyl group is attached to the backbone, it's called NM. So the problem we solved in the previous one was NM, but we said we'll design synthetic ones to see even better models if we can build. So how did we do it, and why did we do it? Our rationale was if you do synthetic molecules, you have less false positives, you can do all possible conformations because in the cells, maybe all sequence combinations are not available if you're only looking at human. Maybe there are some sequence contexts that are not. So how about we generate synthetic ones? So there is a challenge when we do synthetic for nanopore. You need at least 110 nucleotide long molecule to sequence through a nanopore. Most synthetic molecules that you have ever heard, if you have done some chemistry synthesis of RNA, is about 40 or 50 nucleotides. There are very few companies which can generate a perfectly stable, long RNA molecule. And this was part of a major recommendations in a National Academy's report that I was part of on RNA modifications, because we don't have standards for these synthetic molecules. So again, that's again offline topic, which we can talk in good length why. So we used a luciferase mRNA sequence as a backbone because these are established sequences. There is no tying around, structure forming, and all kinds of stuff. And we wanted a molecule to be long enough and have a poly A tail because the protocol for direct RNA sequencing has a poly A. So you can see that the first 30 nucleotides are fixed. Then we add a regular base, right? We added 3 + 3 wobble, random bases, and fixed the middle base. And then we added 20 bases fixed, and then add a GM. This is our design. And then the last 25 base pairs, we added a poly. So this is what we sequenced using flow cells. And you can see clearly that the GM and regular G signals look pretty different. And we started building models. This is only one flow cell. There is a lot of noise. There's not so much data that we generated because it also blocks the pore. I didn't tell you that aspect. These are biological pores. When a molecule moves and it blocks the pore because this moiety is heavy and the area in the pore is not big enough, it'll block. And when it is blocked, it dies. The pore dies. It's a biological one. If you design solid state pores, I was having a conversation with Beth, the future is solid state silicon based kind of pores. So you can wash, reuse, and so on. So your throughput cost and everything will be different. So again, that's a different topic. So we see that from this learning, training, and cross validation curve, we haven't hit it. Okay, there is still room for growth. You can see that, but this is from the nearest neighbors model, and this is a random forest. We didn't like it. No, this was starting to do better. This gives the hope that we can sequence more and get more data to improve that. So where are we going with this? Ideally, we would like to do this for a whole bunch of these modifications. There are about 10, 11 modifications, 19 different modifications currently when I put this slide, which are commercially available. So you can insert them into the design and capture the signals and start building these models, right? This has become a database for the community, right? So, but of course, there are challenges like how do you design without errors? How do you design and we have had challenges where the molecule will fold itself. RNA is not stable, it gets degraded. The company would promise a particular sequence, you won't get that sequence, and you still shell about $2,000 for each design. Each of these designs that you're talking about costs about $1,500 to $2,000. And if something goes wrong, it's a nightmare to work with the data, one, but also the molecule is wasted, okay? So there are some challenges. And again, in the National Academy's report that I was part of, we were talking about who would set these standards, maybe NIST, would start designing these molecules and providing for the community to do. Okay, so I'm sorry, I'm going a bit slow, but yeah. So another area that we have been interested is RNA structure. And RNA structure is important for all life forms because RNA in the cell typically folds like this in a two-dimensional fashion. Of course, it's three-dimensional, but you're seeing a 2D structure. So when it is folded like this in a 2D space, it's called a secondary structure. Now, the secondary and tertiary structure, tertiary is the three-dimensional structure, are known to change dynamically to cellular signals. For instance, secondary and tertiary structure elements can be interconverting depending on the proteins, electrolytes, small molecules, and altering the RNA function. Now, RNA doesn't work itself. It also interacts with other RNA and other proteins. So, okay, cell is a complex sauce. A lot of the things are happening. So, how exactly people solve this structure? Traditionally, people use X-ray crystallography or NMR for solving the 3D structure for RNA, and it's a very time-taking process. But in the last decade or so, people have been fascinated about 2D structure because that's easier. You can do in high throughput. And this is done by a whole bunch of protocols. But all these protocols use the same sauce, which I'm going to elaborate basic sauce. Okay, and we are very in liking of ShapeSec because our collaborator is one of the first who developed this technique, and others followed suit. Okay, so how exactly is the secondary structure of RNA resolved in most of these experimental protocols that I was mentioning? In the cell, the RNA would look like this. Okay, this is the 3D structure of an RNA molecule, and you want to solve the structure. So you would end up throwing a chemical moiety, which would selectively interact and modify the single-stranded region. Okay, this is called the double-stranded region. This is a loop, and this is another region which is single-stranded. So there are chemicals which selectively interact with the single-stranded region. Okay, you can see this. When you throw this, they randomly interact and modify. Okay, again, these are modifications on the RNA. Now, when you do it in a reasonable way, you don't do excess. If you do excess, all these are modified. It gets complex. You do enough. There's something called one-shot kinetics where you throw enough concentration, so each molecule gets one or two modifications. Okay. And our collaborator is very good at Northwestern is very good at doing this. So once you do this modification, you do a reverse transcription. In reverse transcription, your enzyme goes from 3' to 5' and gets the cDNF, which is the basis for all RNA sequencing protocols. Now, when it hits this blockage, this modification, the RT drops, the reverse transcriptase drops off. So when it hits the structure, okay, if it is opened up, it will go through, but the main blockage is this modification itself. Now, when you generate this sequencing data, what you would end up seeing is the single-stranded regions, right? These regions where the blockages are there, you'll see huge spikes in sequencing data because there is a drop-off. But in those regions which are structured, you would end up seeing a pretty neat movement of the molecule, okay? And that is the basis for resolving the structure because you have a constraint to impose on knowing which is single-stranded, which is double-stranded. So experimentally, how this goes is you have an RNA molecule, and you have an untreated. You don't do the reagent. So I'm going to call it as 1M7, which is the reagent that is basically shown in red. And then another side where you don't treat it. Okay, when you don't treat it, the molecule and you do a reverse transcription, you're generating fragments that are shown in green. Okay, you're doing all the reading end to end and getting more molecules of each, and you're sequencing them. So you should expect end-to-end molecules in the control. But when you treat them with this reagent, it will start modifying randomly, right, on these unstructured regions, or what are called single-stranded regions. When these modifications happen, and when you do reverse transcription, it starts blocking and then drops. There is a fragmentation right wherever there is this modification. So you use this neat trick, and then you see a spike. When you compare one versus the other, you start seeing these spikes. These spikes, when you normalize, they're called reactivity scores. Okay, it's showing the extent of reactivity between the chemical that you have used and the RNA that you are interested in. So higher reactivities typically correspond to unstructured regions or single-stranded regions. Lower reactivity scores correspond to structured regions. Okay. Now, most of these efforts have been done using Illumina short-read sequencing technologies. And you can imagine what kind of things can go wrong because you don't have resolution of complete molecules. You're looking at localized regions. And if you have a gene which has 10 different transcripts, you have no clue where the reactivity is high or low, which of these, many of these transcripts have these changes. So we argued, can we use nanopore sequencing for this? Why? because in nanopore there are four bases that are called by the sequencer. But if there are modifications introduced, probably it will tilt the signal. Maybe there is a SNP that is introduced, or maybe we can design algorithms for the current changes. Okay, that is the essential idea. But in the first version, we said we'll use base calling error because we'll trust the nanopore base caller to see if it is calling an error. Okay. Uh, so a little bit about this particular reagent 1M7. Okay, this is the moiety that you throw onto the RNA, and it attaches to the hydroxyl group and adds a bulky fragment. When you add this bulky fragment, when you treat this, it is bound to tilt the signal. Okay, the current. And we noticed that when you use a charged molecule, it tilts the signal even more. Okay. And we played with a molecule which is about 338 nucleotides long. It may appear like short, but you will see in a minute how interesting that molecule can be. And this molecule has been beaten to death in structure, RNA structure. Others have resolved the structure of this particular molecule. So what did we do? We treat this molecule and we do a poly-A, because you need poly-A attachment to the RNA. We do a ligar adapter ligation, prime the flow cell, load the library, and do RNA sequencing. Okay. I'm going fast now because I'm thinking we all are on the same page about nanopore sequencing. So you use this molecule, you do a control unmodified, you do a shape treatment, that is a 1M7, and it would modify in unstructured or single standard regions. You do sequencing of both of these libraries. It's expected that in the shape treated, there will be mutations. Okay, and you end up calling these mutations, and you define what are called as reactivity scores. Okay, if there is a mutation, let's say for a region there are 10,000 reads. At a particular location, 50% of these locations are modified. There's a reactivity score of 50%, 0.5, for instance, right? If a location has only some of the reads supporting a modification, then you will get lower reactivity score and so on. So that's the underlying idea behind. But since this original effort, we started building machine learning models, not trusting the base caller, but doing our own version of the. So we need to define reactivity score. Remember, I said a very simplistic reactivity score. This is again summarizing simple bioinformatics stuff, where you align the reads. If at least the read covers 70% of the region, you keep it. You call the SNPs. You calculate the modification probability along the length. You create a reactivity score based on this. Now, remember, we did a control and we did a treated. The control is we expect nothing to happen. In the treated, you expect changes to occur wherever there's a structure, right? But nanopore also has error rate, right? Remember, this is a technology which is maturing. So you should expect some errors directly from the sequencer, even if you don't do anything. So we tried different versions of reactivity score. The first one is simple x minus y, where x is modification probability at a location in treated, and y is the modification probability in the control, log s, log x, y, all kinds of fancy or simpler models for reactivity score. We realized that the log xy actually worked the best for the reactivity scores. Oh, no. So you're seeing along the length of this 332 nucleotide molecule, the shape treated, control, and the difference. Okay. The difference is plotted as log of x by y. When we looked at all the positive values, those are in agreement with short read based data. That wherever those locations are modified in short read, we noticed that those were the locations that we found. The negative values were a little puzzling for us because they were most likely errors of sequencing. Okay, there's still some error rate because of the. We then used this as a constraint to predict the structure. So this is our inferred structure. This is the gold standard, or people who have repeatedly solved it. This is what we are seeing, and this is based on short read data. And we compared root mean square differences, and we are in much more agreement with this. We are in agreement with this, but we also have changes that we have noticed. And this is just for 330 nucleotide molecule. And imagine if you have a 10,000 nucleotide molecule or something like that. Things can get pretty crazy on how complex the structure. When we started working, my PhD students said, why are we working with 300? Why can't we work with like 2,000 or longer? I said, believe me, you will not like it. And the students spent almost 1 year just analyzing, trying to optimize the parameters and so on. So our idea is to try these on whole cell extracts and do deep sequencing, because since this was done, the cost of sequencing has come down, and there is more excitement of using these long read technologies for mapping. Yeah. How are we doing with time? Okay. Okay. All right, so I'm done with the two parts that I wanted to talk. I'll briefly just give you a high-level overview of what we're trying to do with RNA-RNA interaction mapping. So another thing that happens in the cell is RNA doesn't work alone. They often interact with other RNA or itself. And sometimes these interactions are direct between RNA, and sometimes there's a protein which is facilitating this interaction. Now, this region is called the interacting interface. Just like when you talk about proteins, protein interacting interfaces, RNA have these interacting interfaces. Now, they can interact self, or they can interact with others, or they can interact between multiple molecules. It's a pretty complex history that goes on when we look at the. So there are protein mediated and RNA centric, and these interactions are happening all the time in the cell. For instance, translation, tRNA and mRNAs are interacting, okay? Where microRNAs and mRNAs interact for degradation. The most recent Nobel Prize on microRNA is because people have elucidated how microRNAs work with mRNAs for contributing to the degradation of RNA. And snRNAs and pre-mRNAs are always in interaction in the splicing, in the spliceosomal context. But how much we know about this interaction, how dynamic and all? Pretty little, okay? Because it's hard to map these interactions using at least existing techniques. So again, there's more molecular biology. Almost everything that we do, we try to integrate the protocol with the. I'm going to skip the protein-mediated part. I'm going to focus on the RNA-mediated part, because which is what we have. For mapping these interactions, what typically is done is you use sorrelin with biotin. And sorrelin has the ability to link two pyrimidines in the double-stranded regions. And the biotin has the ability to be able to be pulled down, okay? So in SPLASH, which is a name given to one of the first protocols, people did cross-linking using sorrelin and biotin, and then use beads. These beads have the ability to attract the biotin, for instance, and you start ligating these ends, and one of the ends is left, other is attached, and it's a random thing. That's another topic. You attach the adapters and you do short rate sequencing. Again, the same issues. When you do short rate sequencing, you have a resolution of 50 to 100 base pairs, and you don't know what's the full scale of interaction and the right. So we try to convert, address these challenges by using trying to identify the complete lens and interactions. Okay. Our approach is using nanopore on human embryonic kidney cell line. Again, by now you should have realized that I love cell lines, and we have used HEK293 as a cell line for mapping these interactions. This is the complete flow. Again, I'm going to skip all the details. If you're interested, we can always talk. The end product is mapping RNA-RNA interactions through cDNA sequencing. So in this case, we saw a lot of noise in RNA. Our throughput was low and was getting costly. So we tried to convert the RNA into cDNA, but this was a couple of years ago. Now we're going to try do this on RNA itself. In terms of the bioinformatics protocol, it's a challenge because once you get these long fragments where you expect, so the idea here is, go back. Oh. So the idea here is that these two RNAs are interacting. They may or may not be from same molecule. You ligate them and you sequence. So you have to think that it doesn't have to be 50-50. Okay, there could be 30% of one gene and 70% of the other, 30% of one, 30% of second gene, 30%. It's a very complex interaction. This is a very simplified figure showing two molecules are interacting. What if 3 or more molecules are interacting together? And that's why it's a fairly complicated bioinformatics problem. So we ended up doing a lot of computation, on this data set. So we generated these long reads. Each molecule is an interaction. And we started clustering based on k-mers. Okay? If they have same set of k-mers or significant sharing of k-mer is where k is like 10 or 15 base pairs, we group them into clusters. And finally, when you have grouped them, you still know that a given cluster may have different molecules. Each molecule is an interaction for us. And mapping these reads right, these long reads into different genes is not simple. So you can try 30% alignment, 35% alignment of one gene, and then you keep trying. We use bit score, simple BLAST, okay, bit score and a e-value, a combination of log of that to see what is the local optimal alignment across a bunch of target genes. So that's what was achieved over a period of 4 to 6 months, okay. And the outcome is that about 75% of most of the genes or most of the reads were aligned, okay. This is the median distribution of the coverage, okay, of the finally, although we started with 10 million reads, we ended up with 1.6 million unique clusters. That is still saying 1.6 million interactions, okay? Then we asked how often they interact self. This figure is just showing a small fraction of this RNA or self interaction, and there is an enrichment for mRNAs followed by link RNAs. But when we talk about 2 or more molecule interactions, there is mRNA interactions being the prominent. A small number of them are same mRNA interacting. You can see a slight blue, but most of them are across 2 different molecules. Then link RNA mRNA interactions are the second most popular kind of interactions. And when we extend to 3 molecules or more, you start seeing that still mRNA interactions are popular or most abundant, and the link RNA tries to interact with mRNA, and the same story is repeated. We were also interested in interface. So when you have an interaction, a molecule can interact with multiple interfaces, okay? So the average number of interfaces that we noticed, or median number of interfaces, was about 10. A molecule can have 10 different regions that is interacting. And the number of interfaces only increases as the complexity of interaction. As more number of interactions of a molecule it goes up, there is more number of interfaces that it is using to interact. These are pretty new because we haven't seen any efforts in this space for, yeah. And link RNAs and mRNAs naturally have more number of interaction interfaces because they're longer in length, not surprisingly. And microRNAs and others have lower number of. When we look at the structure of this graph, again, for those of the network enthusiasts, not surprisingly, we would do that because my PhD was in systems biology. We see a scale-free graph, most nodes poorly connected, few nodes highly connected. So we took these highly connected ones and started doing functional enrichment or other kind of analysis. And again, each of these clusters exhibited link RNA mRNA interactions, and there would be like each cluster associated with immune response or something like that, or regulation of cardiac muscle apoptosis. We're still sifting through this data. There's a lot to, and one could be vesicle transport. So my hypothesis is that in the cell there are spatial factories where these RNA interactions are happening, and they're trying to dictate how link RNA, mRNA congregations are going to drive the fate of this RNA, whether it should be in the nucleus, cytoplasm, or even other compartments. This is just, again, a thesis, but we need more work to be able to understand what's going on. And this is just for one cell line and one replicate, but it took us still a lot of time to establish the computational pipeline, and that's something that we will proceed so in the future. So most of the work on modifications was done by Cosina experimentalists, and Doa did a lot of computational work. Some early work was done by Raja Kadmuri. He's now a faculty in India, and Cosina is in the med school. A lot of structure work was done with Julius Lux, is a faculty at Northwestern, and his postdoc. And RNA structure work was done by David. He's a PhD student in the lab. I haven't talked bulk of the work on protein RNA interactions and assays that we have been developing, mapping protein RNA interactions. That is mostly the work of Xander, who is a lecturer and also a PhD student, and Sahiti, who is interested in Plasmodium, which is a malarian parasite for studying RNA modifications. Hunter is interested in imaging, so we used retinal images to predict diabetic retinopathy. That's a totally different area. That's a bit more data intensive. I was debating whether to talk about that or this, and I said, no, I'm going to talk more about genomics than imaging. So I'll stop there. Thanks to all the funders. We have been fortunately funded by NIH, NSF, some collaborative work with some Indian funding agencies, and yeah.